Unit 5 Core Analytical Techniques in Biotechnology. We are going to study one of the very important unit in biotechnology, which is Core Analytical Techniques. These techniques are basically tools and methods that scientists use to study biological samples like cells, proteins, DNA, and many other molecules. Without these techniques, it is very difficult to understand what is happening inside a biological system. This unit is very important especially for exams like GAT-B 2026 because many concepts and application based questions come from here. So in this lecture, we will not only see definitions but also understand how each technique works, where it is used, and what are its limitations in simple and clear way. This slide explains how proper guidance and help in preparing for exams like GAT-B. The focus here is on concept-based learning, which means instead of mugging up, we should understand the logic behind each topic. When concepts are clear, solving MCQs and previous year questions becomes very easy. Also, structured preparation is very important. That means we should have a proper plan starting from registration to final counseling. Regular practice, mock tests, and doubt solving help in improving performance. So overall, smart preparation with proper guidance can give better results. We are covering all major analytical techniques used in biotechnology. First, we will study microscopy, which includes different types like light, fluorescence, confocal, and electron microscopy. Then we will move to centrifugation, where we learn how particles are separated based on size and density. After that, we will cover spectroscopy techniques like UV visible, IR, NMR, and others. Then, chromatography methods like ion exchange, gel filtration, and affinity will be discussed. Finally, we will study electrophoresis and microarray, which are very important for DNA and gene expression analysis. So, this PPT gives a complete overview of analytical tools. Light microscopy is the most basic and commonly used technique in biology. It uses visible light in the range of 400 to 700 nanometers and glass lenses to magnify the sample. The resolution of this microscope is about 0.2 micrometers, which means it cannot see very small structures like viruses clearly. There are different types of light microscopy like bright field, dark field, phase contrast, and DIC. In this technique, contrast is creating either by staining the sample or by differences in refractive index. This is mainly used for studying cell shape and tissue structure, but its limitation is that it cannot show very fine details inside the cell. Fluorescence microscopy works based on special molecules called fluorophores. These molecules absorb light of one wavelength and emit light of a longer wavelength. This difference is called Stokes shift. Because of this property, we can specifically label and detect certain molecules inside the cell. This microscope has important components like light source, excitation filter, dichromatic mirror, and emission filter. It is highly specific and is used for protein localization and live cell imaging. However, it has some limitations like photobleaching, where fluorescence decreases over time, and autofluorescence, where unwanted signals are produced. Confocal microscope is an advanced version of fluorescence microscopy. It uses a laser light and a pinhole system to remove out of focus light. Because of this, It gives very sharp and clear image compared to normal fluorescence microscopy. One of the major advantages of confocal microscopy is that it can produce optical sections and 3D images of the sample. This is very useful when we are working with thick samples or tissues. However, this technique is expensive and takes more time for imaging acquisition, so it is not always used for routine work. Electron microscopy uses electrons instead of light, which have very small wavelength around 0.005 nanometers. Because of this, it gives very high resolution up to 0.1 nanometers, which is much better than light microscopy. There are two main types: TEM, which shows internal structure, and SEM, which shows surface structure. This technique is used to study organelles, viruses, and ultrastructure. However, it requires vacuum conditions and complex sample preparation, and we cannot observe living cells in this method. Centrifugation is a technique used to separate particles based on their size, shape, and density. It works by applying centrifugal force, which pushes heavier particles outward faster than lighter ones. Another important concept is the sedimentation coefficient measured. There are two main types of centrifugation: differential and density gradient. This technique is widely used for separating cell organelles and purifying proteins in laboratories. High speed and ultracentrifugation. High speed centrifugation can reach up to about 25,000 rpm and is mainly used to separate cell organelles like mitochondria. Ultracentrifugation is much more powerful and can go about 1 lakh rpm, which is used to separate very small particles like ribosomes, DNA, and viruses. These are two types: analytical and preparative ultracentrifugation. Analytical is used for studying properties, while preparative is used for sedimentation and collection. This technique requires special conditions like vacuum and temperature control to prevent damage to sample. UV visible spectroscopy is based on electronic transitions in molecules. When light passes through a sample, some light is absorbed, and this absorption can be measured. It follows Beer-Lambert law, which says absorbance is proportional to concentration. This technique works in UV range 200 to 400 nanometers and visible range 400 to 700 nanometers. It is commonly used to measure concentration of DNA and proteins. However, one limitation is that impurities in the sample can affect the accuracy of the results. CT spectroscopy is used to study chiral molecules. It measures the difference in absorption of left and right circulatory polarized light. Since many biological molecules like proteins are chiral, this technique is very useful in biotechnology. This method is mainly used to analyze protein secondary structure. It helps us to understand how much alpha helix and beta sheets is present in a protein. So, it gives important information about protein folding and structure, which is very important for understanding protein function. IR and FTIR spectroscopy. Infrared spectroscopy works by detecting vibrational movements of molecules. When IR radiation passes through a sample, different bonds vibrate at different frequencies. These vibrations help us identify functional groups present in the molecules. FTIR is an advanced version of IR spectroscopy. It uses Fourier transform to collect data faster and more accurately. This technique is widely used for biomolecules characterization and has advantages like high sensitivity and quick analysis. Raman spectroscopy is based on inelastic scattering of light. When light interacts with a molecule, most light is scattered normally, but a small portion changes its energy. This change gives information about molecular structure. One advantage of Raman spectroscopy is that water does not interfere much, so it is very useful for studying biological samples. It is complementary to IR spectroscopy, meaning both together give better understanding of molecular structure. Mass spectrometry is a powerful technique used to measure mass to charge ratio, which is written as m bar z. In this method, molecules are first ionized and then separated based on their mass and charge. Different ionization methods like ESI and MALDI are used in analyzers like TOF helps in detection. This technique is highly sensitive and is widely used in proteomics and metabolomics to identify and analyze biomolecules. NMR spectroscopy is based on the behavior of atomic nuclei in a magnetic field. Certain nuclei like hydrogen and carbon absorb energy and give signals that help us understand their chemical structure. This technique provides detailed information about molecular structure. It is a non-destructive method, meaning the sample is not damaged during analysis. So, it is very useful in structural biology and chemistry. Chromatography is a technique used to separate different components of a mixture. It works based on distribution of molecules between two phases, stationary phase and mobile phase. Different types of chromatography include absorption, partition, and affinity. Important parameters like retention time and resolution help us analyze separation efficiency. This technique is widely used for purification and analysis of biomolecules. Ion exchange chromatography separates molecules based on their charge. In this method, charged molecules bind to oppositely charged groups present on their stationary phase. There are two types, cation exchanges and anion exchanges. Elution is done by increasing salt concentration. This technique is commonly used for protein purification because proteins have different charges at different pH. Gel filtration chromatography is also called size exclusion chromatography. It separates molecules based on their size. The stationary phase contains pores and molecules pass through them. Large molecules cannot enter pores or come out first, while small molecules enter pores or come out later. This technique is used for desalting and estimating molecular weight of proteins. Hydrophobic interaction chromatography. This chromatography is based on hydrophobic interactions between molecules and stationary phase. In high salt concentrations, hydrophobic interactions become stronger, so molecules bind more tightly. Elution is done by decreasing salt concentration, which reduces hydrophobic interactions. This method is useful for protein purification, especially for separating proteins based on their surface properties. Affinity chromatography is a high specific technique. It works based on specific binding between two molecules like enzyme substrate or antibody antigen interaction. For example, his-Tag proteins bind to nickel columns. Elution is done by changing pH or adding a competitive ligand. This method gives very pure product because its high selectivity and it is widely used in protein purification. This slide explains three important types of chromatography. used for different purposes. Gas chromatography (GC) is used for separating volatile compounds, where the mobile phase is gas. It is mainly used in chemical analysis where compounds can easily vaporize. HPLC stands for High Performance Liquid Chromatography, where liquid is used under high pressure for separation. It is very accurate and widely used. FPLC is similar but specially designed for proteins and works under mild conditions so that proteins do not get damaged. All these methods are used for both analytical and preparative purposes. Electrophoresis is a technique used to separate molecules based on their charge and size using an electric field. Which current is applied, molecules move towards opposite charge depend on their nature. There are different types of like agrogel electrophoresis for DNA and RNA, phage or STS phage for proteins. After separation, bands are visualized using staining methods. This technique is very important for analyzing nucleic acids and proteins in research and diagnostics. Microarray is a high throughput technique used to study gene expression. In this method, thousands of DNA probes are fixed on a small chip, and labeled sample DNA or RNA is allowed to bind to them. Based on hybridization, we can detect which genes are active or inactive. This technique is widely used in genomics and disease diagnostics. It helps in analyzing large amount of data at one time, making it very powerful in modern biology. This slides gives a quick summary of all techniques. In microscopy, we studied light, fluorescence, confocal, and electron microscopy, each with different resolution and application. In centrifugation, separation is based on size and density using centrifugal force. In spectroscopy, UV visible is used for concentration, CD for protein structure, IR and Raman for functional groups, MS for molecular masses, and NMR for structure. Chromatography separates based on charge, size, and affinity. Electrophoresis separates DNA and proteins, and microarrays is used for gene expression analysis. This slide is very useful for last minute revision. This slide emphasizes the importance of staying updated during preparation. Regular updates, tips, and guidance can help students stay on track and improve their performance. Consistent effort with simple preparation and strong concepts leads to better results. So students should keep revising regularly and stay connected with reliable study material. Subscribe Brainzz for more videos. The final slide highlights complete preparation support, including notes, MCQs, mock tests, and doubt solving. It shows that structured learning and regular practice are key for success in competitive exams. It also emphasizes the preparation should be simple but conceptually strong. With proper guidance and consistent effort, students can achieve good scores and succeed in exams like GAT-B.